Hey guys! In the previous dot point, we did a lot of stuff on what enzymes are and their role in metabolism. In this dot point, we will be exploring why chocolate is the meaning of life, as well as identifying pH as a way of describing the acidity of a substance. Okay, the first part was a lie. There is no talk about Snickers bars today. In layman's terms, pH is a measure of acidity and is measured by using the pH scale. The pH scale ranges from 0 to 14, has more colours than a rainbow, and measures the number of hydrogen ions in a solution. The lower the number on the pH scale, the more acidic a solution is, and the more hydrogen ions it has. Conversely, the higher the number on the pH scale, the more basic a solution is, and the less hydrogen ions it has. A pH of 7 represents a neutral solution, meaning that the number of hydrogen and hydroxide ions are equal. For the curious folk out there, acidic solutions taste sour, basic solutions taste bitter, and neutral solutions are things like water and don't taste like anything. Whilst today is generally focused on learning what pH is and the pH scale, we're also going to look at the relationship between enzymes and pH. Each enzyme has an optimum pH level. This is important, so note this. As you can see on the graph, the blue dot indicated here represents the optimum pH level. This is where the enzyme activity is highest and where the enzyme can most effectively catalyze chemical reactions. Just like how having too much or too little chocolate in one sitting is generally bad. When the pH level increases or decreases from this blue dot, enzyme activity has decreased and efficiency of a chemical reaction is lowered. When a solution is too acidic or basic, the extreme pHs will cause the denaturing of the enzyme. Denaturing means that the structure of the enzyme is altered and changed because the extreme pH levels break the bonds between the polypeptides, especially the ones that create active sites. Since the active site is changed, the enzyme can no longer do its job, and there is very minimal or even non-existent enzyme activity. Basically, the enzyme is damaged and ruined. Chemical reactions are now slowed to a crawl, and no amount of chocolate will fix that. This can be summarized using a simple flowchart. Extreme pH levels, or something that is too acidic or basic, will cause a reduction in enzyme activity. Less activity means less efficiency, and the cell therefore suffers and can stop functioning properly as a result. Let's have a look at an example. Urease is an enzyme used to speed up the chemical reaction that breaks down urea to form carbon dioxide and ammonia. That previous part is just for the curious and completely unimportant to what we're doing, but this next part is. As you can see here, urease works best at an optimal pH level of 6 to 7. As the pH increases or decreases from this optimum, there is lower enzyme activity. At the extreme end, the enzyme has become denatured and there is low enzyme activity as a result of this. And there you have it. We're done. Let's quickly go over the concepts we've looked at today and then you're free to go get yourself some chocolate. Firstly, pH is a measure of acidity. We measure this through the pH scale. Secondly, there is a strong relationship between enzymes and pH. Enzymes have an optimal pH range where the enzyme works most effectively. When the pH of an enzyme is too high or too low, the enzyme could be denatured and therefore have minimal and very low enzyme activity. In the next dot point, we will come full circle and explain why pH is an important factor in enzymes. There will also be no further talk about chocolate.